Hey, Mr. P here.
What's up, guys? It's Mr. Schmitz.
In this video, we're going to start our investigation of DNA structure, and we're going to kick it off with how DNA was discovered in the first place.
So, believe it or not, we didn't always know that DNA existed in the cell. And furthermore, we didn't know what it did.
Yeah.
But through a couple of experiments, we actually started to figure out what it is and what it does. Let's take a look. Let's take a look.
So the first one that we're going to focus on is Frederick Griffith, and he kind of kicked off our current understanding of DNA using a variety of techniques that we
still use today, right?
Yeah. And so he had a big question. So all research projects start with a research question. And so he said, how do bacteria cause pneumonia? At the time, pneumonia was a really big deal. They didn't know what the cause of it was. Is it magic? Is it viral? Is it DNA? Is it protein? Is it chemical? Is it a toxin? We don't know. So he set out to figure out what it is that actually caused pneumonia in the bacteria.
In doing this, he was able to, using mice, isolate two different strains of the bacteria pneumonia. So in these two strains, only one of the two of them was harmful, is what we would call it, or actually caused the illness that we think of as pneumonia, even though they're both strains of the virus, or the bacteria itself, excuse me.
Yeah, and when you look at each of them, they both grow very well in the Griffith lab. He had plates of both. Both of them appeared to look exactly the same. They were growing on the same media. They could be inoculated into the same host. However, when you did inoculate the bacteria into the same host, which in this case was lab mice, they caused very different results. There is an S strain and there is an R strain. S stands for smooth and R stands for rough. It basically talks about their appearance. When the S strain was inoculated into a mouse or injected into a mouse, it killed the mouse, caused pneumonia. Mice cannot recover from pneumonia in this case, and so it killed the mice. When the R strain bacteria were injected into the mice, it kept them viable and they did not contract pneumonia. So right off the bat, two strains of bacteria, one resulted in pneumonia, one did not.
So with that information, he's able to take it a step further. And he used a process called heat killing, which is basically he heated up the pneumonia to a temperature that they were no longer alive, but still had sort of their pieces intact, I guess. And he injected that, and what he found was that obviously the mouse survived, it remained healthy, and that there were no bacterial growth that occurred.
And basically the result was it killed the bacteria, and he proved that it killed the bacteria because no bacteria grew, and it didn't kill the mice. And so when he took the heat-killed bacteria, which he proved when it was isolated, and he mixed the heat-killed S with living R, notice that the living R kept the mice healthy and the dead S kept the mice healthy. And when he mixed those together, living R, heat-killed S, he injected those into mice. Presumably, he thought that they would keep the mice healthy. They contracted pneumonia. It killed the mice. And then when he further investigated the bacteria that caused pneumonia in this mouse, he concluded that both R and S colonies were isolated, which means one of two things. Either the dead S came back to life, or the living R changed into the living S. So what made the first group sick?
Pneumonia? What are we getting at here?
So, when looking at the Griffith experiment in more detail and kind of putting it all together, drawing conclusions from it, what made the first group sick? Did the S bacteria, the living S, did they produce a toxin or a poison that killed the mice? Or was it the fact that they were present that killed the mice? When Griffith applied heat and killed the S bacteria and injected them into the mice remained healthy, suggesting the cause of disease was not poison.
Right, so it's something living in the bacteria or the bacteria itself being alive, because when you inject the dead version, it doesn't harm the mice.
Yeah, and the reason he employed this particular trial was because if the bacteria produced a toxin and you killed the bacteria, the toxin should still be present, right? And it didn't produce any kind of disease, and so it can't be a toxin. He basically solidified his understanding of that particular question.
That'd be like a control group, right?
Yeah, control, experimental. Right? So, when Griffith injected a mixture of the heat-killed S and the living R, the mice developed pneumonia. That was the other big discovery. Okay? When he examined the lung tissue of these dead mice, he determined the living cells had grown inside. Again, like I alluded to on that first slide, how can this happen if the S cells are dead?
And so this gets to like the real crux of his experiment is how is it possible for us to like create a new version of a cell, right?
Yeah.
And so what did he conclude?
Mixing the two types of bacteria allowed a chemical factor to transform the heat-killed S-type cells into the living R cells. So he is concluding that there has to be a changing from the living R to the heat-killed S. He coined the term transformation because he said that there has to be some kind of chemical compound that contains information that tells the bacteria how to do its job, in this case be pathogenic, and it changed the harmless bacteria into disease-causing ones, presumably killing the mice at the conclusion of that swap.
So there's something that contains information or instructions that could be passed back and forth.
We will just say, at this point, he knows that there are instructions. There is a chemical compound. It's an entity. It's an actual thing containing instructions that has to be transformed between two species of bacteria to cause disease.
I'm sure you guys on the other side of the screen are shouting the answer of DNA right now, but remember, we don't know that at this point in time. This is part of this discovery process.
Yep. So what does this transformation process look like? Basically, you have dead bacterial cells. Remember, this is the heat-killed S, and you have the living bacteria cell, or the living live R.
Both were harmless.
Both harmless, right? If you kill this and you keep this alive, both of those harmless, don't do anything. But in this mixture, there is a situation in which DNA fragments, these are called plasmids, can actually be taken in by the living cell. It's basically this dead bacterial cell passing the baton to this living bacterial cell. It causes the living bacteria cell to start replicating the compound, which in this case is DNA. It makes it have a set of instructions it didn't have to begin with, which in this case is cause pneumonia, be pathogenic. And as soon as it starts reading that and it becomes part of its own genome, it's a transformed cell. In this case, it would transform into the live S, and it can cause disease. So, since the ability to cause disease was inherited by the offspring of the transformed bacteria, I conclude that the transforming factor has to be a gene.
And that is a key term here, a gene being a sequence of DNA that codes for a protein, which we will get more in depth into genes as we talk through DNA and genetics.
Yep, so we've started with a compound. We know it has instructions. Now we're saying it is a gene, and so we're getting closer to that discovery of DNA.
Absolutely. So again, that living bacteria is accepting those instructions that he talked about from the dead, integrating it into its own genome, and utilizing it as a new trait.
And we're going to learn later, but we know that now, after discovering DNA, that DNA contains genes. These genes code for proteins. Proteins code for traits. In this case, disease-causing.
Right. So, Griffith's experiment sort of paved the way. It began the process of thought for some other scientists. But Griffith's experiment alone didn't prove the existence of DNA or the function of DNA by itself. And so some scientists had to sort of take the baton and move it further, which brings us to our next experiment completed by Mr. Avery as well as some of his counterparts.
Yeah, and so he being Avery and his colleagues, looked at the work being done by Griffith. Griffith never used the term DNA, never got to the result that we know today, that it was DNA. And so he basically said, we know that there's genes, we know that there's information, we know that there's a molecule that codes for that, but...
Which molecule?
But which molecule? What does it look like? We have no idea. We haven't seen it. We need to find it. And so he employed this particular experimental protocol, and he said only one possible conclusion could explain these results. DNA was the transforming factor. So he created a hypothesis. He said that it has to be DNA. We're going to go about trying to conclude that. And
so in his experiment, he hypothesized it was DNA, and it was sort of a debate between is it the proteins or the DNA and which is responsible. Because at the time, we knew that there were proteins and we knew that there were DNA always present in and around the nucleus, but we didn't know which was sort of responsible for which part of this process.
Yeah. So he took a culture. It has a bunch of these cells growing in it. It's just a liquid medium with bacteria cells in it. He centrifuged it, basically just made it more concentrated so he could get to the cells. He basically had two different experimental trials that we want to worry about in this particular class. He used anti-protein enzymes and he used anti-DNA proteins. We know from our previous units that enzymes typically end in ase, And so if he took this particular sample and treated it with protease, protease is an enzyme that breaks down proteins. So when he treated the particular sample with protein,
removing enzymes,
you're going to not kill the bacteria. Transformation will occur, and you will get both R cells and S cells. And so he drew a conclusion from that the active factor cannot be protein, because if you remove the proteins, you still had transformation.
So he was still using the same strains of pneumonia that
Right.
Griffith was using.
Yep.
And he was just testing to see, is it still disease-causing if I remove the proteins or not?
Yeah.
And when he removed proteins, like Mr. P just showed you, we still have that disease-causing bacteria.
Yep. So isolated protein couldn't be it. Transformation still happened. So he took it a step further, and now he treated with ribonucleases, which basically takes away the RNA, and noticed that transformation still occurred, and so the active factor is not RNA in this case as well. And so the only logical thing after that, if we take the protein away, if we take the RNA away, is to take the DNA away. Deoxyribonuclease is an enzyme that breaks down DNA. Employed that particular enzyme, breaks down the DNA, no transformation occurred, and that is only going to mean one thing, and that is the active factor has to be DNA, because if you take it away and transformation doesn't occur, then that is the result.
Yeah, so to oversimplify, he went through 3 trials. He removed the protein in one trial, the RNA in one trial, and the DNA in one trial. And the only time it killed the cell or prevented transformation from occurring was when he removed the DNA. And that's that result that he got, proving that DNA is responsible for genes, which is what Griffith began this process saying.
And so at this point, conclusion, DNA is the cause of transformation.
And that brings us to our third and final experiment completed by Hershey-Chase. These two worked to solidify that conclusion and prove it once and for all by using a little bit different technique called a bacteriophage. Mr. P, what do we got here?
And so in this particular experiment, he had 2 different trials. He did an experiment 1 where they were testing proteins, and there was an experiment 2 where they were testing DNA. Again, we wanted to conclude whether it was DNA or protein that caused disease. In this experiment, we used bacteriophage viruses. So this is a viral infection instead of a bacteria infection. It's a viral infection of a bacteria cell, but still talking pathogenicity, still talking disease-causing agents. Is it protein or is it DNA? And so he took 2 different radioactive isotopes. He took radioactive phosphorus, and that was used to radioactively label the DNA. He used radioactive sulfur, which is going to radioactively label the protein coat, because viruses are basically made of 2 things. You have a capsid, which is a protein coat, and you have DNA kept within that head. So if you have an agent that has DNA and protein exclusively, you can basically isolate one and test it. So this radioactive sulfur is going to radioactively label this protein coat. He then allowed the bacteriophage to infect the bacteria cell. It injected the DNA, which did not contain the radioactivity. He put it through a centrifuge protocol, and he found that most or all of the radioactivity resided outside of that bacteria pellet, meaning the radioactivity stayed within that protein coat, which was not injected into the bacteria cell, resulting in a conclusion that protein cannot be the genetic factor because the protein wasn't even injected or ever entered the cell.
Right, so a bacteriophage invades a host by injecting something to take over control of that cell. In this experiment, he found that they didn't inject the protein.
Yeah, protein stayed outside.
So what did they do in the other version?
Now you use radioactive phosphorus. Phosphorus is one of the components of DNA. And so when you radioactively label phosphorus, you link it to the viral DNA. You let the viruses do the things that they're supposed to do, which is inject that DNA into the host cell. Now the radioactivity is within the host cell. Notice it's inside. If you centrifuge again to isolate the bacteria from the viral protein coats, you'll notice that all of the radioactivity is now in that pellet, and that is a conclusion. You can draw a conclusion that DNA has to be the genetic material that the viruses are using to hijack the host cell, which can be seen when looking at that result.
So this experiment solidified our understanding once and for all that it is truly DNA that is responsible for the instructions that we call genes and the passing on of those traits. So we, through these 3 experiments, learned what DNA was responsible for within the cell.
And hopefully now you have a greater appreciation for DNA and how it was discovered.
Thanks so much for being here.